actions at the tissues, the role of the receptors. There are two distinct types of receptors, the membrane-bound or G-linked protein receptor and the steroid or DNA binding complex receptor. Regardless of which type of receptor is being utilized, it is the receptor that will ultimately determine the action of the hormone at the target tissue. The G-linked protein receptor functions by activating a secondary messenger within the target cell, while the steroid receptor will directly go to the nucleus where it will act as a binding or regulatory complex. The membrane-bound receptor are receptors that are utilized by the non-steroid hormones. These non-steroid hormones activate secondary messenger sequences. These secondary messenger sequences include cyclic AMP or a PIP2IP3 and calcium-mediated activated sequence. Either way, there is a kinase activation that follows the activation of the secondary messenger. This activation sequence alters the mRNA transcription and protein synthesis, all of which is based on what type of kinase has been activated. Therefore, we will see a distinct response based on both the hormone and the stressor that elicits the activation of the various pathways. In this pathway, we're going to look at the cyclic AMP activation. What we have is we have the activation of the G-linked protein. This activation of the G-linked protein will activate adenylyl cyclase, which activates a cyclic AMP. By activating a cyclic AMP, we either activate the response element or a kinase. The kinase changes cellular function by itself. The change in cellular function and the cyclic AMP activated response element both go to the nucleus. This change in the nucleus leads to an increase in mRNA, tRNA, and RNA transcription rates, all of which lead to an increase in protein synthesis. It's this increase in protein synthesis that leads to a greater change in cellular function, which will then inhibit the cyclic AMP signaling cascade. Eventually, the hormone is released, and the cell goes back to normal function. Here we see the overall sequence of events laid out as a single flow. Now what happens with the PIP2IP3 pathway? We have the same initial activation sequence. However, all of the G-linked proteins will initiate a lipase activity. This will initiate a conversion of PIP2 to IP3, which causes the release of calcium from the endoplasmic reticulum. This release of calcium from the endoplasmic reticulum will activate the protein kinase and thus cause a change in cellular function. The change in cellular function leads to a change in mRNA, tRNA, and rRNA sequencing and transcription rate, which leads to a change in protein synthesis, and this change in protein synthesis leads to additional change in cellular function. It's the additional change in cellular function that leads to a sequestration of the calcium back in the endoplasmic reticulum. Additionally, the conversion of PIP2 to IP3 frees up what's known as a DAG, a diacylglyceride. This DAG can activate the protein kinase in a different fashion than the IP3 and calcium can. It can also act as inhibitory on the IP3 signaling towards calcium release from the endoplasmic reticulum. And here we see the activation sequence laid out. The second type of receptor is going to be the steroid receptor. This is for the lipid-soluble steroid hormones and does not involve a G-linked protein. The binding of the steroid hormone to its receptor will activate a DNA regulatory complex. This will couple with the regulatory enzymes in the nucleus to form a DNA regulatory element. This element will initiate an increased mRNA transcription and protein synthesis. Some steroid hormones will actually inactivate the process of mRNA transcription and thus will diminish the amount of protein synthesis. There will also or can also be secondary kinases activated by the steroid hormones, but we will not discuss that here. We will simply look at the regulatory complex on the DNA. What ends up happening is the steroid hormone will first become free from its binding globulin, at which point in time it will diffuse across the membrane and bind to its receptor complex. The receptor complex will then become activated by dissociating a heat-sensitive protein, HSP, from the inhibitory center on the receptor complex, forming the steroid hormone complex. This steroid hormone complex will then head into the nucleus, where it will bind with the DNA binding regulatory protein to form a regulatory element. This regulatory element will then change the mRNA, tRNA, and RNA transcription rate, leading to a change in protein synthesis and thus a change in cellular function. This change in cellular function will head back to the nucleus, inhibiting the regulatory elements and causing a dissociation of the steroid hormone from its receptor complex, while at the same time inhibiting additional protein synthesis because the cellular function has already changed. And this is what the whole schematic will look like as a single flow. As stated earlier, the regulation of the receptor is the actual means for controlling the action of the hormone. What does that mean? In actuality, it's the receptor that will ultimately determine how much activity the hormone might have for the body. The response of the receptor at the target tissue is going to depend upon the concentration of the receptor, which is modified based on the metabolic stress, as well as the concentration of the hormone. There are going to be three distinct responses that we will see here. We will see an upregulation, a downregulation, and what is usually referred to as an insensitivity to the hormone at the target tissue itself. Upregulation. Upregulation is a process by which more receptors are embedded into the membrane of the target tissue. This is typically done when the cell requires a greater stimulus with limited amount of hormone being present in order to accomplish the necessary alteration in functions. It could also be seen when there is an increase in stress within the cell without a change in hormone within circulation. Overall, the upregulation will induce a greater response or an ease by which the cell is triggered for the hormone signal. And let's look at a cartoon of the process. Cell at rest. cell is stressed. When the cell is stressed or when the body senses a disturbance, hormones will be released in response to the help signal from the stress. At the target tissue, there will be membrane receptors embedded into the membrane, which will start to interact and induce a hormone-initiated response. This hormone-initiated response is going to attempt to change cell function. However, that cell function is not enough to eliminate the whole entire stress. Within the cell will initiate an upregulation moment where more receptors will be embedded into the membrane in an attempt to get additional response by interaction with the hormone. Which leads to no more stress being felt and an appropriate change in cell function. In response, the cell will have additional receptors already embedded within the membrane so that it will be ready for the next stress signal. Opposite of the upregulation is the downregulation process. In the downregulation process, the cell will remove receptors from the membrane. This is typically done when the cell does not have the indicated stress for which the hormone is being released or has experienced overload from the signal of the hormone within its normal function. Overall, downregulation is an attempt to induce a less robust response or where it can become generally harder to trigger a response by the hormonal signal. So let's look at a cartoon of downregulation of the receptor. cell at rest. We get a stress signal that the cell does not sense. In response to the stress signal, hormones are released into circulation. But once again, the cell does not sense the stress. We have the interaction between the hormone with its receptors, and initiating its response, attempting to change cell function in order to return homeostasis from the stress. However, the cell does not respond because it does not have the stress. This change in cell function leads to an overworking stress within the cell itself. In an attempt to get the cell itself back to homeostasis, the cell will remove membrane receptors in an attempt to diminish the amount of hormone-induced response that it's having. Because it was overworked due to the last hormone signaling sequence, the cell will diminish the amount of receptors that are visible on its membrane for that particular hormone. Insensitivity. Insensitivity is where we have inducing a lower level of response at the cell without changing the total number of receptors within the membrane. This is typically done when the cell is experiencing a stress that is contradictory or inhibitory to the indicated stress for which the hormone has been released. And this leads to an inhibition at the receptor or within the activation sequence of the receptor. The overall effect within the body to this insensitivity is usually an increase in the concentration of the hormone within the plasma. This is not seen with the other types of regulations. So let's take a look at the cartoon of an insensitivity of the receptor. Once again, we have the cell at rest. We now have a stress signal. However, the stress signal for the body does not meet the stress signal within the cell. Hormones are released. In an attempt to limit the amount of response that we see from the hormone, the cell will inhibit the activation sequence at the receptors, minimizing the amount of hormone-initiated response, allowing for only small amounts of cellular change. The small amounts of cellular change is because of the inhibition caused by the internal stress. Because there is coexisting stresses that are counterintuitive to each other, the cell itself starts to have an overwork stress. This overwork stress ultimately leads to the cell shutting down its response altogether to that hormone. Things to think about. What type of receptors do non-steroid hormones interact with? What is the difference between the mechanism of action for the non-steroid and steroid hormones? How does stress change the amount of a hormone receptor that is present on the membrane? How is the modification of receptors the real regulators of hormone action?